everyone, and thank you for joining us today for a webinar on unlocking urinary cfDNA insights, where we will be discussing a complete workflow from sample collection to detection. My name is Claire McLean, and I'm a marketing associate here at Omega Bio-tek, and I'm joined today by two of our experts, Dr. Kieran Dervasula, one of our product managers, as well as DNA Genotek's senior R&D manager, Mike Tabb. On the agenda for today, I'll start off by giving you a brief introduction to Omega Bio-tek before handing you off to Mike for an introduction to DNA Genotek as well as their Colli-P system. From there, Kieran will walk you through our cell-free DNA extraction workflow as well as showcase some results from our ddPCR application. Finally, we'll end today's session with a Q&A with our experts, so please submit your questions as they come. With that, I'd like to introduce you to Omega Bio-tek. We were founded in 1998 and are based out of Atlanta, Georgia, and our expertise is in providing DNA and RNA purification solutions. We boast a portfolio of over 400 products encompassing different extraction technologies, including magnetic beads, silica spin columns, filter plates, and salt-out methods. Omega Bio-tek is ISO 9001 and 13485 certified, and our presence is felt around the globe through more than 50 distribution partners. At Omega Bio-tek, our purpose is to deliver nucleic acid purification solutions that simplify your project and meet your unique needs. Your success is our top priority, and we empower our customers by making automation and quality products more accessible and affordable. With that, I will hand you off to Mike Tabb from DNA Genotek.
Thanks, Claire. As Claire said, my name's Mike Tabb, and I'm a senior R&D manager leading the product and application support team at DNA Genotek. DNA Genotek's located in Ottawa, Canada, and we're a subsidiary of OraSure Technologies. The OraSure family of companies strives to improve access, quality, and the value of healthcare by innovating with effortless tests and sample collection solutions. We do this through offering a diverse portfolio of diagnostic rapid tests through OraSure and through our sister company, Sherlock Biosciences. And by providing a broad suite of sample collection devices from DNA Genotek. And today I'm going to focus on the sample management solutions portion of our business. So on this slide, I'm showing a comprehensive overview of all of the product offerings that DNA Genotek has for human research and clinical diagnostics from our molecular solutions segment. We specialize in sample collection products across the human genomics, microbiome, metabolomics, infectious disease, and oncology spaces. Through our portfolio of collection devices, DNA Genotek aims to increase access to healthcare, and we do that primarily by enabling individuals to self-collect their samples. And they can do that either at home or in a clinic setting. Our devices preserve samples by preventing microbial growth and preventing sample degradation using some unique stabilization chemistries. Our devices greatly reduce logistics costs by eliminating the need for cold chain shipping and storage. And on the right-hand side of this slide, I've got an image of our Colli-P urine collection device, and that's the device that I'm going to talk about more throughout this presentation. And since I'm going to be focusing on urine collection devices, I'd just like to briefly touch a little bit on urinary cancers. So in the last few years, there's been almost 1,000 articles published demonstrating the utility of urine in cancer research. And it may be intuitive that certain urogenital cancers, such as urothelial cancer, prostate, bladder, kidney, it may be intuitive that all of those can be detected in urine. It's also been shown that biomarkers from other cancers, such as lung and breast cancer, can be detected in urine as well. And I'm showing some of those additional cancers in the figure on the right. And what this really speaks to is the enormous potential of urine as a sample type for early detection of cancer. And I've got a few examples of those publications here. There's publications for detection of prostate cancer in urine, some other cancers like endometrial cancer, as well as some non-urogenital cancers, such as hepatocellular carcinoma. So for urine biomarker detection, it's often preferable to collect what's known as first void urine. And sometimes first void urine is also called first catch or first pass urine. And although the name implies it, first void urine does not necessarily refer to the first urine passed in a day. But rather it's typically defined as up to the first 50 milliliters of urine that is passed during any urination. And it can be collected at any time of day, so it's not specifically collected in the morning. And the reason that first void urine is of particular interest is because it's highly concentrated with biomarkers specific for infectious disease and oncology applications. So using first void urine may help ensure that you have the best sensitivity for any downstream assays that you might want to run. Here I'll talk a little bit in a little bit more detail about the Colli-P urine collection device. And let me start by saying that Colli-P is the only first void volumetric urine collection device on the market. We've designed Colli-P to be user-friendly for all genders, and it really enables hygienic and completely non-invasive self-collection of samples. And there's been a number of clinical trials that have shown that Colli-P is actually preferred over other collection methods by the users. And unlike traditional urine cups, Colli-P allows users to collect a standardized volumetric sample, and they can do that without interrupting the urine flow. And there's different versions of the Colli-P device available, and those allow you to collect a range of urine volumes. You can collect as little as 4 milliliters or up to a 40 milliliter sample. And that really allows accommodation of a range of assays with different input requirements. The sample tubes provided with the device, and I show some of those on the image on the right-hand side, they're designed to be compatible with automated sample processing platforms. And the collection tubes that come with Colli-P are pre-filled with a preservative that stabilizes the analyte content of the urine, and it enables shipment and storage at ambient temperature. So the major benefit here is that you get significant reductions in logistics costs associated with sample collection and sample transport. And importantly, we keep the volume of the stabilizing solution in Colli-P low compared to the volume of urine being collected in order to prevent over-dilution of the sample. Here I have a short video that demonstrates how the Colli-P device works. So the user uncaps the tube containing the preservative and attaches it to the Colli-P collection device. As you'll see here, Colli-P is designed with flexibility in mind, and it allows the user to collect in both standing and seated positions. The user urinates into the device funnel and the urine is channeled into the collection tube until the designated volume is collected, at which point the remainder of the urine is directed into the toilet. There's no need to interrupt the urine stream. After collection, the user detaches the tube and recaps. At this point, the sample is stable at ambient temperature and is ready for transport back to the lab. As mentioned previously, many urine analytes are concentrated in the first void fraction of the urine. For that reason, it's critical to control the volume of urine collected in order to prevent overdilution of the analytes. This results in a reduction in assay variability due to noise and ensures that the assay sensitivity is not negatively impacted. The ColiP device provides minimal variability in the volume of urine collected when you compare it to traditional urine cup collections. With urine cups, participants tend to overcollect, resulting in a more dilute sample. The chart on the right here shows the volumes of urine collected using different ColiP device formats compared to urine cups. So as you can see, the volume of urine collected with ColiP is very close to the target volume for each ColiP device. And the target volumes are shown across the bottom of the x-axis on this chart. And what you'll notice is that the volume variability with ColiP is very low. The gray bar on the right-hand side is a traditional urine cup collection. And in this case, participants were instructed to collect a 20 ml first void fraction of urine. As you can see, people had a difficult time stopping the collection at the 20 ml mark, and they tended to over-collect. Additionally, the volume variability across the participant group was much higher than with ColiP. I'll talk a little bit about the urine stabilization ability of ColiP as well. As I mentioned, it's designed to stabilize the analytes in urine. So analyte degradation begins right at the time of collection, and signal from certain analytes degrades rapidly when the sample is not stabilized. If urine is held unpreserved at room temperature, which you'll often see happening during transport or if there's delays in processing, several changes can happen quite rapidly. Microbial growth occurs, and that leads to contamination of the sample with microbial DNA. And additionally, you can have physiochemical changes such as changes in pH or changes in the concentration of certain analytes, as well as lysis of host cells in the sample. To minimize the types of changes that happen post-collection, ColiP devices are available with two different stabilization solutions. They're called UAS and UCM. So the UAS stabilizer is designed to preserve cell-free DNA and extracellular vesicular RNA for up to 7 days at room temperature. So this really allows you, again, to eliminate the need for cold chain transportation and storage. Additionally, UAS is non-lytic, so it maintains urine homeostasis. So urine collected using ColiP UAS is suitable for use in oncological liquid biopsy applications. And the chemistry itself is non-lytic in nature. So what that does is it preserves the extracellular space and it reduces the introduction of noise due to contamination with cellular material. The other stabilization solution, UCM, is designed for general preservation of collected urine samples. Potential application areas here include infectious disease testing, such as for HPV and STIs, And there are a lot of research studies and clinical trials that are currently generating interesting results in this space. And it's important to note that both of these stabilization chemistries are safe for consumer use. So this really enables self-collection at home. On this slide, I have a couple of charts showing some data that demonstrates the ability of ColiP to stabilize cell-free DNA and maintain homeostasis in urine. So on the left, I show some data demonstrating cell-free DNA stability. So in this study, participants were asked to collect first void urine into a ColiP device with and without added UAS stabilization solution. The cfDNA was extracted from samples at baseline, which is just shortly after collection, as well as after subjecting the samples to either simulated transport conditions, which in this case consist of a number of freeze-thaw cycles between -20 Celsius and 40 degrees Celsius. And also after storage for 7 days at room temperature. And here we used a qPCR of a common housekeeping gene, beta globin, to look at cfDNA stability. And as you can see, cfDNA was successfully stabilized in ColiP UAS samples, as indicated by the delta CT of near zero, and the UAS stabilized samples are shown in blue here. And in contrast, the unstabilized urine, which is denoted by no addition on this chart, shown in red, exhibits significantly higher delta CTs, indicating significant loss in signal from cfDNA after both simulated transport and room temperature storage at 7 days. On the right-hand side, I have some similar data demonstrating ColiP's ability to maintain homeostasis in the sample. In particular, here we look at urinary host cellular stability. In this case, urine was collected in ColiP devices, again with and without the UAS stabilizer, and then it was centrifuged to obtain cell pellets. And this was done both at baseline and after the same simulated transport and storage conditions mentioned previously. We extracted and measured the amount of genomic DNA in the cell pellet. And in this case, if cell integrity is maintained, you would expect to see no change over time in the amount of DNA recovered from that pellet. But if cell lysis is occurring, you'd expect to see a loss of genomic DNA. So here we show that cell integrity is maintained in the ColiP UAS samples, as again shown by the near zero delta CT values shown in blue. And in contrast, the unstabilized urine in red exhibits significant loss in the qPCR signal, suggesting a large loss in host DNA from the cell pellet due to cell lysis. And I'll conclude my portion of the webinar here. If you'd like more information on ColiP urine collection devices, please reach out to us. Our contact information is here, and it'll be shown again at the end of the webinar. And with that, I'll turn the talk over to Kieran from Omega Bio-tek. Thanks, everyone.
Thank you, Mike. Hi, everybody. My name is Kiran Durasula, and I'm going to spearhead the part 2 of the webinar focusing on harnessing liquid biopsies for clinical implementation. Urine-based liquid biopsies have emerged as non-invasive, cost-effective, and scalable alternatives of blood-based diagnostics. However, to realize their full clinical potential, important considerations must be allotted to 1, pre-analytical preservation. This has been expertly covered by Mike in the earlier part of this talk, and it is a crucial factor to protect the integrity of our analyte of interest. 2 is appropriate, reliable extraction methodology. Unlike plasma, cfDNA from urine is often low yielding, hence protocols and processes that are optimized for maximizing cfDNA yield with high specificity, reproducibility, and automation is essential for real-world implementation. And third but not the least, appropriate downstream technology. Recent advances in molecular techniques such as next-generation sequencing and droplet digital PCR have enhanced the specificity of extracting or analyzing cfDNA from various bodily fluids. So choice of the right platform is something that one would need to make to be able to draw relevant conclusions with high degree of precision for downstream of interest. With that, the objective of the study today is to provide our audience with the proof of concept solutions that encompasses these three different factors. We've validated a complete workflow solution from sample collection to analysis. The urine sample was collected and stabilized in DNA Genotek's Colli-P UAS. And cfDNA was extracted from the preserved urine sample using Omega Bio-Tek's extraction chemistry. And subsequently, the purified cfDNA was used in a droplet digital PCR downstream for detection of rare variant alleles. Before I move on to the crux of the study, I would like to address why cfDNA is so difficult to extract. cfDNA is transient in nature and present in various bodily fluids. It's short, around 170 base pairs, and fragmented, and has a very short half-life, often ranging from several minutes to few hours. And also, cfDNA is found in low concentrations, and it also depends on different factors such as depends on specific individual circumstances, for example, normal versus disease, and even the extraction methodology that is used to extract cfDNA. The amount of cfDNA that is extracted is also influenced by the sample input volume. And in some cases, larger sample input volumes are needed to be able to extract this less abundant cfDNA to quantities that are required to be used in your downstream of interest. And studies have actually shown that various commercial kits that are employed actually result in varying amounts of cfDNA. Hence, the recovery rate is a crucial factor and has a significant bearing on the success of your subsequent analysis. Hence, accurate methods are important, and it's important to establish reliable methods to isolate cfDNA that is not only higher yielding, but can also accord high sensitivity and specificity to your downstream assay. To address this need, Omega Bio-Tek has launched a magnetic bead-based extraction kit that can isolate circulating cell-free DNA from plasma, serum, and urine. And this kit is called the MagBind cfDNA kit, and it's available as a RUO and even in a CE-IVDR format. And since this kit is magnetic bead-based, it's a scalable, and the entire protocol is automation friendly. And honestly, the entire protocol can be automated depending on the liquid handling platform that you're using. As I mentioned, the scalability factor, so this kit can handle sample volumes ranging from 1 mil to 10 mil. And Omega Bio-Tek's unique chemistry uses lower lysis buffer volume and binding volume. So we can actually automate and process the entire 10 mil volume without sample splitting using appropriate plasticware and appropriate liquid handling platform. Our chemistry can elute cfDNA in volumes as low as 50 microliters, providing our end user with highly concentrated cfDNA. This kit is also available in other plug-and-play formats. On the left here, this kit is available in a pre-filled seal cartridge format, which can be used on our MagBinder Fit 24 magnetic processor. And it can process 48 4-mil samples. And the processing time for this kit on MagBinder Fit 24 is going to be around 50 minutes online time and a 30-minute offline lysis time. And again, this is for 24 samples. On the right here is our pre-fill sealed reservoir option for our Hamilton Star and other liquid handling customers. All the reagents are available in sealed format, and this can process 96 4 ml samples. And on Hamilton Star, this protocol can be completely automated and can be processed in around 3.5 hours. I would like to go over the experimental overview of this study. The first void urine sample from anonymous female donor was collected and added to DNA Genotek's Colibri UAS at a ratio of 2.3:1. To mimic a clinical sample, we have purchased cfDNA reference standards from Horizon Discovery and spiked in 350 nanograms into the preserved urine sample. So these cfDNA reference standards come as a set of 4 vials, and they contain 8 onco-relevant mutations at different allelic frequencies. So the 4 vials, each of them, the allelic frequencies range from 5%, 1%, 0.1%, and 0%. So the 0% allelic frequencies are 100% wild type, and that serves as our control. So we have 4 experimental groups for this particular study. Group 1, which is our experimental condition, where each of these spike-in standards were individually spiked into preserved urine samples in UAS. And we have 3 different controls. Control 1 is our no spike-in control. So this is just going to be our preserved urine in UAS. This is to make sure that none of these mutations are present in our sample itself. The control 2 is our no urine, no preservative control. So it is just the spike-in that we purchased in elution buffer. And this is to ensure that we are calling the right mutations and we are seeing the expected allelic frequencies that are being advertised by the manufacturer. And the third spike-in control is our no urine control. So it's just the reference standards in UAS. This is to ensure that there is no interference of UAS in our downstream analysis. So 4 ml of sample from each of these experimental groups was extracted using MagBind cfDNA kit using a modified protocol. And we eluted cfDNA from all these experimental groups in 50 microliter volume. So the purified cfDNA was actually put through a barrage of tests. We performed tape station analysis of the purified cfDNA to look at the cfDNA profile as well as the yield that was obtained from each of these experimental groups. And the purified cfDNA was also employed in a downstream droplet digital PCR workflow. And we assayed for NRAS and EGFR gene variants and looked at different allelic frequencies. The tape station analysis of the purified cfDNA from all of these experimental groups is depicted on this slide. As you can see, we have dark banding pattern between 150 to 200 base pairs. That is where you expect the cfDNA to be present, and we see that across all the experimental groups. And if you all focus your attention to our no spike-in control, so this is just our preserved urine in UAS. We do see the cfDNA band and a dark band which indicates gDNA present in the urine sample. So this is just the endogenous cfDNA and genomic DNA that's present in the collected urine sample. And as you would expect, you see that gDNA band in the preserved urine sample with spike-in as well. And the other 2 experimental groups, which has no urine, you did not see that band. So this kind of reiterates that this band is emerging from the urine sample itself. So we used the regional functionality analysis of the tape station software, and estimated the cfDNA yield from each of these experimental groups. So the group which has no spike in, just the preserved urine sample, the endogenous cfDNA yield was around 58 nanograms. And if you look at the groups with either just spike in and elution buffer or spike in and UAS with we had a yield of around 161 to around 186. So with a 350 nanogram input to each of the sample, the recovery rate was around 46 to 53%. And in our experimental group where we spiked in the reference standards to preserved urine, the yield was actually higher, which makes sense because here we have the endogenous cfDNA from urine as well as the cfDNA from the spike in sample. So we actually had pretty good recovery across all the experimental groups. So moving on to the droplet digital PCR assay that we performed, this slide, we're looking at the allele populations in the NRAS mutation assay. Each assay generated around 15,000 droplets. And the pink horizontal line that you see, that's the threshold that was manually set by using the 100% wild type as the control. And we ensured that there is good separation between the positive and negative droplet populations. And if you focus your attention on the no template control group, there are no droplets. So this indicates and confirms that there is no non-specific amplification. And if you look at our no spike in control group, as well as our 100% wild type group in the mutant assay, there are no positive droplets, which is as expected. And we see this good positive droplet population across all the allelic frequencies that we've tested in all the groups where there was spike in. So we see a similar story with our EGFR assay as well. So looking at the allele populations, again, we set the threshold manually looking at the 100% wild type control. And again, we did not see droplets and then no template control indicating no non-specific amplification. And as expected, we did not see positive droplets in our no spike in control, as well as our 100% wild type in the mutant assay. And positive droplets were detected across all the allelic populations, allelic frequencies that were tested. So by tabulating the positive droplets, we performed this concordance analysis between the observed allelic frequency and expected allelic frequency. So since these are reference standards, we have information about the expected allelic frequency, and the table down below is actually the range one would expect when you spike in these standards. And this is based on the acceptance criteria based on the manufacturer's certificate of analysis. And the graph on the left, you see the concordance analysis graph with an R square of 1. So and we see good concordance between our observed allelic frequencies, and they're pretty close to the R square equals 1 line. And what this tells us is that we are able to detect mutant alleles at frequencies as low as 0.1%. And all the other observed allelic frequencies are in excellent concordance with expected frequencies for all the experimental groups tested, and it falls within that the expected allelic frequency range that was set by the acceptance criteria based on the spike in CFA. So we did a similar exercise with the EGFR assay as well. So we tabulated the positive droplet population to perform the concordance analysis between the observed versus expected. And we are able to detect the mutant alleles as low as 0.1%. All the experimental groups except for one at the higher end, we see excellent concordance. That There's just one experimental group where the 5% allelic frequency was spiked into urine. We detected slightly lower. It was around 2.5, where the acceptance criteria places it between 3.5 to 6.5. But again, the lower end, we are in excellent concordance. With that, I would like to move on to my conclusion slide. The major takeaways from the study are as follows. So optimal collection and preservation conditions, as well as high sensitivity, reliable cfDNA extraction, are integral for successful downstream implementation. And the complete workflow solution that we have provided is pretty efficient. It's calling mutations at the right allelic frequencies. And we observe excellent concordance between the expected as well as the experimental groups. And our sample collection to extraction workflow is capable of detecting mutant alleles at frequency as low as 0.1%. With that, I would like to acknowledge Carissa Townsend, research scientist at Omega Bio-tek, for carrying out the experimental work. And I would also like to acknowledge Emory Integrated Genomics Core for their contributions towards the droplet digital PCR work and analysis. With that, I thank you all for your attention and pass the baton back to Claire. Thank you all.